Hello everyone, my name is Ibrahim and I am a PhD student in Dr. Varadharajan Sundaramurthy's lab at NCBS. Today I will present an overview of my thesis entitled Mycobacterium tuberculosis sulfolipid 1 induces TRPV4 mediated lysosomal rewiring and drives macrophage cell death. Tuberculosis disease caused by Mycobacterium tuberculosis (MTB) remains one of the leading cause of death from an infectious disease worldwide. MTB primarily infects macrophages, the immune cells that are responsible for engulfing and eliminating invading pathogens. However, MTB has evolved sophisticated mechanism to survive and replicate within these cells. Previous work from our lab made an interesting observation. MTB-infected macrophages actually exhibited a marked increase in lysosomal biogenesis and lysosome activity. It was further identified that one of the major MTB cell wall lipids, sulfolipid 1 (SL1), was sufficient to induce this lysosomal expansion through activation of TFEB, the master regulator of lysosome biogenesis. However, the upstream signaling mechanism linking SL1 to TFEB activation remained unknown. To address this question, we first investigated the role of calcium signaling. We found that extracellular calcium influx is essential for the SL1 induced lysosome response, as removing extracellular calcium significantly impaired lysosome biogenesis. We next identified the calcium channel TRPV4 as the critical mediator of this process. Both pharmacological inhibition and genetic depletion of TRPV4 markedly reduce the SL1-induced increase in lysosomes, demonstrating that TRPV4 is required for this process. Importantly, activation of TRPV4 alone was sufficient to increase lysosome biogenesis and lysosome activity, indicating that TRPV4 is not only necessary but also sufficient to drive lysosome remodeling. Furthermore, TRPV4 activation enhanced the delivery of intracellular MTB to lysosomes, suggesting an important role in regulating host-pathogen interaction. Beyond lysosome biogenesis, we discovered that SL1 also promotes lysosomal exocytosis, and this process likewise depends on TRPV4 activity, revealing that MTB extensively rewires lysosomal function. Finally, to understand the broader consequences of this lysosomal remodeling, we performed transcriptomics analysis. RNA sequencing analysis revealed significant upregulation of lysosome-associated genes following MTB infection. Integrative analysis across multiple infection datasets identified a key lysosomal modification that is strongly associated with macrophage cell death during MTB infection. Overall, our study uncovers a previously unrecognized signaling pathway in which the MTB virulence lipid sulfolipid 1 activates TRPV4 mediated calcium influx, leading to the lysosomal rewiring. These findings provide new mechanistic insights into host pathogen interaction and identify TRPV4 as a potential therapeutic target for tuberculosis. With that, I would like to sincerely thank my supervisor, Dr. Varadharajan Sundaramurthy, my co-supervisor, Dr. Sai Subramanian, all the members of our lab and our funding agency for their continuous guidance and support.